In this recording, I would like to go over the slides that you were asked to view in your virtual histology lab that you performed last week. And I'd like to highlight some key things that you should be focused on as you go back and review both the chapter and the drawings that you made for this exercise. So let's start with epithelial tissue. Epithelial tissue commonly is going to line the outside of the body and surfaces inside the body that communicate with the outside, as well as covering the surfaces of organs and the interior cavities of the body, like the pleural cavity or the pericardium or the abdominal cavity. And let's start, therefore, with what are called simple squamous epithelial cells. And here in this particular slide, you can see we're looking down upon the cells. They look irregularly shaped. They look like pavers that are linked together, forming a walkway. And you can see the central nucleus very evidently. In some of the slides you looked at, though, you would have seen this from a side view, and you would see why they're called simple squamous. They're very, very flat. Their job is to allow easy diffusion of materials into and out of those cells. So they act as a barrier, but they want to allow things to move in and out. I've highlighted in the past, you would find simple squamous cells, for example, inside the alveolar sacs in your lungs. They have to be very thin so that they can facilitate diffusion of oxygen across that barrier between the air that's contained in the sacs and the gases that are dissolved in the blood. So oxygen-rich air in the sacs needs to get into the blood, so the oxygen needs to diffuse through there very readily. And going in the opposite direction, the CO2 that has built up in the blood as cells have metabolized and producing CO2 as a byproduct, we need to get that into the air sac so we can exhale that. So very flattened types of cells and very characteristic for where you want easy diffusion. You've seen this also, another key one that readily comes to mind are the walls of blood vessels, like in the capillaries, where you've got the lining of the blood vessels made up of simple squamous cells. A second kind of epithelial tissue are simple cuboidal cells. All right, and here looks like we probably have tubules that are found in the kidneys. You can see that we have cross sections of multiple tubules. There's almost a dozen tubules here that have been cut across, and you can see the cells are not nearly as flat as we saw with the squamous cells. Very prominent nuclei. They're surrounding the lumen, the actual duct itself, where things that are going to be absorbed or things that are going to be secreted are going to travel to. These tubules are bunched up against each other, and it looks like between some of the tubule walls you can see some blood vessels, maybe blood cells, red blood cells in those blood vessels, those capillaries, providing nutrition to these living cells. So wherever you want secretion and absorption to take place, obviously a very highly desirable feature. In the kidney tubules, you're going to have simple cuboidal cells. So let's go on now. A third type are columnar cells. Now these are elongated. Here it looks like probably we are viewing villi or microvilli in the small intestine. The cells are more elongated. You can see the nuclei are pretty prominent. So where absorption is really important, you're going to have columnar cells. Hard to tell in this particular section whether we can see some of the modified columnar cells, the goblet cells, that would be producing mucus that would help line these epithelial cells so they are not damaged by any digestive enzymes or by hydrochloric acid in the lumen of the small intestine. So wherever you want absorption taking place, most prominently, probably in the GI tract, you're going to have columnar cells. Okay, now moving on to connective tissue. There are various connective tissue samples that you viewed. One in particular was bone. And here you see in this slide a very iconic depiction of bone. You can see that we have, looks like, I don't know, 3 or 4 osteons or haversian systems, either wholly or partially depicted in this particular slide. So if we zero in on the central part of this slide, very prominently in the very center of that haversian system, you have the haversian canal, and that is how you're going to have innervation. You're going to have nerve endings in there, and you're going to supply nutrition. There are going to be blood vessels traveling through there. So that haversian canal with its concentric lamellae that you can see radiating out from that central position, you can see it's kind of layered. It looks almost like you cut across a tree trunk and you're seeing the concentric rings in a tree trunk. And they are kind of demarked because there are clefts between the lamellae where the osteocytes, the actual bone cells, are going to reside. And so the lamellae, the bulk of the lamellae are actually made up of a matrix that is going to become impregnated with minerals like calcium to strengthen, to provide the structural integrity of the bone. And so those osteocytes, they're kind of stranded in those lacunae, and so they are able to communicate with one another, radiating out from the osteocytes. You see the canaliculi, and that's the way that they are going to communicate with one another. So you can tell just the way this thing is set up that it's pretty dense, pretty compact. A second type of connective tissue are fat cells, adipose tissue. Again, a very iconic depiction here. Maybe this is fat that's deposited around some of the internal organs, the abdominal organs. You can see individual cell is mostly made up of fat, adipose. It looks empty, but that's, that would have been washed out when they stained this. The fat would dissolve away. But that's where the fat would reside. And so the nucleus of these adipocytes are pushed up against the actual cell membrane. Great storehouse for energy, obviously. This is where excess calories are being stored away in the form of fat. Moving on, then, we have what is called loose connective tissue or areolar connective tissue. You can see this does not look nearly as dense. You see lots of fibers. Those are collagen and elastic and reticular fibers crisscrossing throughout the background matrix, which is almost, we refer to it as a ground substance. It's almost in a fluid state. You can see all kinds of cells that are spread throughout here. The smaller ones are going to be the fibroblasts that actually are producing those fibers that I just mentioned. But you're going to have other things. You're going to have parts of the immune system. Some of those larger ones are macrophages and mast cells. Areolar tissue, key place that you're going to find that is in the subcutaneous layers of the skin, kind of forming the base on which the epithelial cells of the epidermis are going to reside and are going to attach to the body. So you'll find this around a lot of organs in the body, and you can see it helps offer some support. A third type of connective tissue you looked at was cartilage. This particular slide happens to be elastic cartilage, and there are a variety of different cartilages, but they typically are found at the ends of bones, covering the ends where bones coming together, where they articulate. We don't want them grinding against each other, so they offer a lubricated joint which they can relatively smoothly move past one another. Here, again, you kind of see these little clefts, these lacunae, and the cartilage cells, the chondrocytes that are going to actually produce the cartilage are found inside of those lacunae. This looks quite a bit denser than you saw with the areolar tissue. But, variety of places that we've talked about where you would find cartilage, this would be the type of cartilage that would be present. And funny as it seems, blood is considered to be a type of connective tissue. Here you can clearly see all sorts of red blood cells. Remember, they're biconcave, they're enucleated, they don't have any nuclei in them. And then you see some other components of blood, for example, the platelets that allow clotting to occur. You have white blood cells. Here we have a couple of different types. We have monocytes, neutrophils, lymphocytes, and they tend to have very characteristic profiles, which would make them easily identifiable. When we talk about the circulatory system, we can talk about the particulars, the number of lobes you look for. You can see that just on inspection here. They have different number of lobes. It allows them to be identified from and distinguished from one another. Moving on to a third type of tissue, of course, is muscle. There are three types of muscle: skeletal, smooth, and cardiac muscle. Here we're seeing skeletal muscle, skeletal indicating these are going to be found connecting different parts of the skeleton, the bones, allowing for movement of body parts and movement of the entire person, whole body. Here you see very characteristic striations. So those striations represent when we go down to an even finer level, a lower level of structure, that we would actually have the proteins that make up the muscle tissue, the actin and myosin that overlap, that are going to ratchet as they connect to one another, that allows muscles to shorten. And then, of course, if we break those interconnections between the actin and myosin, then we can allow muscle fiber to elongate, like when you have an extension. So contraction, we're shortening them; extension, we're stretching them out. You can also see pushed up against the sides of the tissue of the cells are the nuclei of the particular skeletal muscle fibers. Again, a very iconic depiction of skeletal muscle. Smooth muscle, which we would find in organs like throughout the digestive tract, to move food and move materials through the stomach, small intestine, large intestine. We have muscles in the walls of those structures. Here they're more independent from each other. You can see a whole array of them. The striations are not as obvious here. And these are going to, when they contract, we have what's called peristalsis. That's the movement of food and digestive materials through the GI tract, and their function is to facilitate that movement. Again, very prominent nuclei present here. Okay, and you can see they're kind of, you sort of get the sense that they are tapered. I mean, if we went to a higher magnification, probably the ones that you looked at in your virtual histology lab, you could see the tapered cells. And the third type, of course, are the cardiac muscles, and these are going to be found in the heart. You have a lot of interconnections. In class, several times I've mentioned how we want these acting in unison. If you're thinking of your left ventricle, which is the chamber of the heart that's going to pump the bulk of the blood through your entire circulatory system, we need them to act in unison. We want to empty that as much as we can to get a good volume of blood flowing. And you can see nuclei, but you can also see those little cross hatchings. Those are the intercalated discs that allow an action potential to travel from one fiber to the next so that we have that coordinated contraction that we desire. All right, so that's cardiac muscle. And then finally, the fourth type of tissue is the nerve tissue, and here is the key component, obviously, of nerve tissue are the actual neurons, the cells, and this is a great slide because it shows very evidently the fibers that lead into the body of the neuron, the dendrites, and then you have that elongated exiting fiber, the axon, which is going to allow this neuron to communicate either with maybe a muscle, maybe this is going to innervate a muscle cell and get the muscle cell to contract, or maybe with a subsequent neuron so that we can have the message, the electronic message passed from one cell to another along a particular pathway, maybe in the central nervous system, in the brain, or in the spinal cord, and this one looks like it's probably a motor neuron. We'll see later, of course, we've got sensory neurons that when we talk about, for example, in the skin, in the integumentary system, we've got sensory fibers that are underneath the epidermis, down in the dermis, down in the support tissue, the connective tissue underneath. That we're going to have specialized cells that can detect pressure and temperature, and that's how we interact with our environment. Of course, that's how we're going to pick up information.